And welcome to the Science Lab. My name is Natalie Brett and I am the 7th and 8th grade science teacher. And yes, I'm coming to you virtually. However, my absence is in the name of love. Right now I'm at a wedding and I just had to be there because I'm the matchmaker. In fact, my childhood friend and my dear friend from college fell in love. So I'm there to celebrate with them. In fact, right now, I'm probably giving a toast, so thank you for understanding as to why I could not make it there this evening. But I'm going to give you a rundown of what to look forward to. We're going to talk about curriculum projections. I'm going to explain my assessment weighting. I'm going to overemphasize the fact that the students do have a cumulative final exam this year, and yes, it does count. And then we're going to talk about Sycamore, our new computer program / our new middle names. And then I'd love to answer any questions y'all have. Feel free to email me and I will get back to you as soon as possible. So first quarter, we've started already with lab safety, basically what not to do in the science lab. Right now the students are preparing for their first formal lab report, which is their Sprouts lab. We're very excited. It's a way of applying what they know about the scientific method, so they'll be coming up with their own manipulated variable. They'll be gathering data based upon the responding variable, putting that into a format, and then communicating their conclusion to the rest of us, their scientific community. So we will probably begin that on Monday Fun Day. Then we're going to switch gears. Chapter 2 is all about what is life. Essentially, it's the classification system. We have an acronym to remember this. Did King Philip come over for good seafood? You probably recall this from growing up. Domain, kingdom, phylum, class, order, family, genus, species. So we'll be focusing on the levels of classification, and then we're going to look in detail on how those units are organized. We're going to start with the cellular level, tissues, organs, and systems. We'll begin talking all about cell functions, essentially cells and their organelles. We'll have a neat hands-on project so the students can demonstrate their understanding of the functions of all the different organelles within plant and animal cells. And then, boom, second quarter. Second quarter, we're going to focus a little bit more in detail on the cellular processes, so specifically photosynthesis, respiration, fermentation, cell division like mitosis and meiosis. And then we'll switch gears again and begin to focus on genetics. And then right around Christmas, we'll come back and we'll spend some time on Charles Darwin and the theory of evolution. And then right around spring break, we start to talk about marine biology, make y'all's trips to the beach a little bit more interesting. We'll begin with super primitive organisms. We're going to talk about sponges, like SpongeBob. There are actually real sponges in the ocean. And then we're going to talk about cnidarians, which are jellyfish and coral. And then we're going to talk about worms and the 3 different groups of worms: platyhelminthes, which are flatworms, nematoda, which are roundworms, and annelids, which are segmented worms. And that will be our very first 7th grade life science dissection. We'll be dissecting an earthworm. And then we will jump into 4th quarter, which is mollusks. And that includes gastropods, which are slugs and snails, bivalves and univalves. So bivalves includes like scallops, clams, oysters, mussels. And then we'll look at cephalopods, which are squid, octopi, nautilus, and cuttlefish. And then we'll focus on arthropods and the major groups of arthropods, which are crustaceans, anything crusty, I like to say, crabs, shrimp, lobsters, things like this. The second group, which is arachnids, spiders, ticks, mites, and scorpions. And then we'll look at centipedes and millipedes, and we'll finish up our arthropod unit on insects. And this will be our second dissection of the year. We'll be dissecting a grasshopper. And then we will look at echinoderms. Echinoderms, there's a song that goes with echinoderms, and it sounds like this. Echinoderms have spiny skin, spiny skin, spiny skin. Now you'll never forget them, echinoderms. And yes, they do have spiny skin. Things like sea urchins, sea stars, sea cucumbers. And then we'll switch gears again. We'll talk about fish. To prepare for our third dissection, we'll be dissecting a perch fish. But we're going to begin with primitive organisms called jawless fish. They're classic naphtha, hagfish, and lampreys. And then we'll look at Actinopterygii, which are those bony fish. Finding Nemo is my favorite example of a bony fish. And of course, be looking at the anatomy of a bony fish via this perch fish dissection. And then we'll finish up with the third group of fish, which are class Chondrichthyes, which is cartilaginous fish. So basically sharks, skates, rays, and chimeras, which is like a hybrid between a shark and a skate. And then we'll have our fourth dissection, which is going to be a spiny dogfish shark, and essentially be able to compare and contrast Actinopterygii to Chondrichthyes, bony fish to cartilaginous fish, look at their similarities and differences. And then we're going to focus on some of the organisms that are more familiar to us. So we'll be working our way up complexity-wise, and we'll begin amphibians, so frogs and toads and newts and salamanders, and we'll get ready for our fifth dissection, which is a grass frog, and then we'll be looking at reptiles, snakes, lizards, turtles, crocs, gators, no dissections, and then birds and mammals. And then at the end of the year, to prepare for our field trip to the Chattahoochee Nature Center, we'll be studying plants, Kingdom Plantae. We'll be looking at monocots versus dicots. We'll be looking at gymnosperms versus angiosperms, and this will be the student's last dissection. We'll be dissecting an angiosperm, which is just a fancy way of saying a flowering plant. So they have lots of fun things to look forward to, obviously in a short amount of time, but not to fret. The students will be over-prepared for their final exam. Yes, it is over all of these 11 chapters. Speaking of weighting, real quick, the assessments are weighted as follows. Tests are weighted fairly heavily, 35% of their final grade, and they do have one chapter test per chapter, so 11 of those heavily weighted tests. And then projects are 25% of their final grade. Those are fairly heavily weighted as well, and we will be doing lots of different projects and hands-on activities. I mentioned the cells and their organelles project, but we'll also be doing some interesting projects throughout the year, specifically based upon systems of the human body. We'll be talking about pathogens and how the immune system works to fight against the pathogens, and they'll be assigned to a group project, a virus brochure. So make sure y'all wash your hands before you leave here this evening. And then labs and participation, basically labs, their formal lab reports are weighted 15%, quizzes are 15%, and their quizzes are announced, and they do know ahead of time when those assessments, formal assessments will be. And then homework is 10%, and my homework policy is as follows. Every quarter, the student starts with a 100. Every time the student does not do their homework, it's -5, and you cannot work your way back up. So if you don't do your homework twice in one quarter, you have a 90 for 10% of your grade. And I do not check for accuracy. I simply just check for effort put forth. If they attempt the homework, then they will get credit for it. And then, let's see, Sycamore, yes. If the student is absent or is missing a piece of paper or didn't finish taking notes because they had to leave and come back for whatever reason, everything they need is on Sycamore. It's under the Documents tab, and they can find all the homework assignments posted there, and they can find my entire PowerPoint presentation there, basically all my Bellwork slides, which have the homework due dates, the project due dates, and what to look forward to, so the quiz and test dates. Those dates are also posted on the calendar, which is another tab of Sycamore. If y'all haven't explored Sycamore yet, I highly encourage you to do so you can familiarize yourself with the interface. However, I took a screenshot of what my class course looks like. It's on the next slide, so if you're curious, you can flip to that at the end of your time in the science lab. And then last but not least, my favorite method of reviewing is called Clever Catch, and some of you may be curious as to what that is. Thank you. This is Clever Catch. So it's for my kinesthetic learners, the kids who learn by moving or doing. Essentially, wherever their finger lands, they have to ask themselves that question. These are questions from the study guide. These are questions from the test. But the good news is it works. And y'all can make this at home. So an old soccer ball, an old basketball, an old volleyball, y'all can manipulate it so that it has questions to prepare the student for the formal assessment coming up. Any resources that you may need will also be on Sycamore. Thanks for your time. Enjoy the rest of your day. Good luck circle swimming. Bye. Smell you later.